most exciting part about finding a species that we thought was extinct is that then we can actually try to conserve it. Then we actually have an ability to protect what's still there and potentially do captive propagation or reintroduction to put it in places where it used to be but aren't anymore.
From the heart of The Ohio State University on the Oval, this is Voices of Excellence from the College of Arts and Sciences with your host, David Staley. Voices focuses on the innovative work of arts and sciences faculty and staff, with departments as wide-ranging as art, astronomy, chemistry and biochemistry, physics, emergent materials, and mathematics and languages, among many others. The college always has something exciting happening. Join us to find out what's new now.
I'm speaking this morning with Nathan Whelan, Associate Professor in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University College of the Arts and Sciences. He serves as Director of the Museum of Biological Diversity, which we're going to be talking about this morning. Dr. Whelan, welcome to Voices.
Thanks for having me.
Well, and let's start with the Museum of Biological Diversity. What would you say is the core mission of this lab?
The museum here at Ohio State is really a research collection. We don't have a public-facing exhibit, so most of what we do is focus on research, although we are trying to get more into the community since I've started. But really what we do is support research on organismal biology, a lot of conservation research, and we have several collections that are strengths of our museum, including the mollusk collection. We have a great entomology collection. We have
bugs.
Bugs. Herbarium, which is plants. We have a tetrapod, so those are things like birds and mammals collection. And we also have a bioacoustics collection, which is unique. So these are recordings of biological sounds like birds and insects.
So why these collections? Maybe I should start with that. What's the purpose of the collection, I guess?
Yeah, I mean, the purpose of the collection really is to document the natural history of our world, so the organisms that are out there that were historically out there. We have some strengths of our collection about the animals that are here in Ohio, the animals and plants here in Ohio. So, for example, our fish collection, we have at least one specimen of every native fish from Ohio. We also have a lot of specimens, especially in our mollusk collection, of specimens from Ohio that have unfortunately gone extinct or have been extirpated or gone extinct specifically in Ohio. So these are records of the historical biodiversity of the state and of other areas in the world, too. So we can use that to understand current biodiversity in the context of what used to be there and what is still there.
Comparable to any other institutes in the world?
I mean, I would say that there's a number of universities and other institutions that have collections that are about our size. University of Michigan, for example, has a similar collection. In terms of size, University of Florida, my expertise is mollusks, so I know a little bit more about mollusk collections. But our mollusk collection here at Ohio State is probably one of the most scientifically valuable mollusk collections in the nation, both in terms of size and importance of specimens. We have some of the last collected specimens that were ever collected here in Ohio of species that are now extinct.
So why mollusks? Of all the things in the world to study, why mollusks?
I mean, that's a good question. One of the things that fascinates me about mollusks is how understudied they are. I think they need an advocate, and part of that is because they're so important to the healthy functioning of our rivers and streams here in the U.S. and Ohio. If we have a good population of freshwater mollusks, so mussels and snails, then we can probably expect that river to be pretty healthy. And, of course, water is very important for society. We need that to function. We need fresh water. We use water every single day in our lives. That water, although it comes from our faucets, it really comes from rivers and streams in the areas. And so making sure that we have healthy mollusks can help make sure that we have drinkable, healthy water.
Now, I know one of your research focuses, maybe your most important, is to prevent the extinction of these mollusks. Say a little more about what the danger is, I guess.
Yeah, so mollusks are really sensitive to changes in water quality and habitat. And so that's one of the reasons why, if we have a healthy mollusk population, we're likely to have a healthy river because they're very sensitive to pollution. They're really sensitive to habitat modification. So when a dam goes in, that can modify the habitat and cause species decline. So a lot of what my lab and my research does is oftentimes using genomic data to assess populations and to better understand species and understand what can be done to conserve these animals, but also we use genetic data to evaluate conservation programs. So things like hatchery propagation and habitat restoration to kind of see how these mollusks are responding in real time or a few years later to conservation actions.
Say a little more about genomic data. I know this has become really important in biology.
Yeah, so when I started in grad school in the mid 2000s, we were sequencing a single gene here and there, and the lab work was actually rather time consuming. Now we can sequence whole genomes or a subset of the genome of a single organism very rapidly and for relatively cheap. So that's enabled us to do a lot of more fine scale analyses because the data is higher resolution if we have a whole genome rather than a single gene. So we can look at things like what's called demographic modeling, so understanding population size through time. That can tell us a little bit about what happened before humans were affecting the ecosystem, and then we can also use genetic data to look at, for example, population size, or what's called effective population size, which is essentially the number of individuals contributing to the next generation. So that gives us a sense of how healthy a population might be. And having that higher scale genomic data really allows us to do things that we couldn't do 10 years ago. And that's especially true for these really understudied organisms.
How is this data collected?
Yeah, so we go out into the field a lot of times, or maybe we'll have a partner. We work a lot with different agencies like U.S. Fish and Wildlife Service, Ohio Department of Natural Resources. And so the first thing we do is we go out and we sample these individuals. A lot of the sampling we do is non-lethal. So one of the things that my lab developed was non-lethal methods for getting genetic material that could be used for genome scale analyses in freshwater mussels. So what we'll do is we'll go out there, we'll find a mussel, we'll kind of pry it open a little bit. We don't think it really hurts the animal. And then we stick a little swab into the animal and swab its foot. This is very similar as if you went to the doctor and had a swab on the inside of your cheek to get genetic material for yourself. Maybe you've done like a 23andMe. It's very similar to that in the way that we get that material. And then we bring it into the lab, we do some lab work, and then then it's ready for sequencing. And typically we send sequencing off to a core facility, and then we'll get gigabytes and gigabytes worth of data. We do a lot of computational biology to process that. So that's also a big part of this work.
Just a quick definition of computational biology.
Computational biology is just really any kind of biology that's taking place, using computer. So analyzing the data. When you have gigabytes of data, you've got to kind of sift through it and figure out, these are the variable parts of the genome that are actually going to tell us something about the biology of these organisms. And then we use computational tools, different programs to actually analyze things like genetic diversity of a population. We can look at how individuals and populations are related, which can tell us a little bit about how we should maybe approach conservation of the organisms.
So when you say collection, when you say museum collection, is it a collection of data or is it a collection of like actual squishy things?
Yeah, it's a collection of actual squishy things, or in the case of mollusks, hard shells. We have a lot of, actually one of the strengths of our museum is that we actually have a lot of soft bodies in our mollusk collection. I'm talking a little bit about mollusks because that's kind of my background, but this is also really unique to the Ohio State Museum of Biological Diversity, is that a lot of mollusk collections just have the shells, or maybe they'll have a handful of soft bodies preserved. But our collection has a lot of soft bodies, and it's curated in such a way that we can track the soft body directly back to the shell. And so that's an additional resource to kind of understand these animals. And we're actually thinking right now about ways to use those soft bodies for research. They've kind of been, I think, maybe overlooked, especially on the snail side. And so we're trying to understand or think about how we can use that incredible resource to better understand the biology of these organisms. But then we also have, insects that are pinned to shells. A lot of that's kind of how insects are preserved. Our fish are mostly in chemical preservatives like ethanol and formalin. And so depending on the organismal type really depends on how the organisms are preserved. But at the end of the day, these are collections of actual specimens that you can go and feel and see. And in some cases, we can actually even get DNA from specimens that had been collected, 20, 50 plus years ago, which is another huge resource.
I know a lot of your work focuses on generating information that can improve conservation outcomes. Give us an example of this sort of work.
Yeah, so a lot of conservation work right now is focused on, obviously preventing extinctions, but one of the things that has been done for a while with fish and is moving into other organisms is captive propagation. So this is happening, at things like zoos, but also at hatcheries, so state-run hatcheries or national fish hatcheries, or we even have a mussel facility out here with Ohio State that's a partnership between Ohio State, the Columbus Zoo, the Ohio Department of Natural Resources, and one of the things that conservation practitioners are doing right now are propagating lots of mussels and thinking about putting them back into places where they used to be, but for some reason the population declined, they're not there anymore. and we want to put them back to restore the habitat. So a lot of times if we think about habitat restoration, we might be thinking about restoring a riverbank or taking out a small dam. But another part of that is actually restoring the organisms that used to live there. Mm-hmm. So one of the things that goes into that is making sure that we're propagating in a hatchery genetically diverse individuals. We don't want to release individuals that don't have any genetic diversity or a population that doesn't have any genetic diversity. So one of the things that my lab has done in the past is evaluate hatchery programs, evaluate the methods that we're using for example, with freshwater mussels to make sure that we're actually generating genetically diverse captive cohorts that can be put back in the wild. And why that's valuable is because generally genetic diversity is seen as giving an organismal or an organismal group a better chance of long-term survival.
I hope this isn't impertinent to ask. Are you in effect designing an ecosystem?
Well, I think a lot of time, I wouldn't say we're designing an ecosystem. I would say that we're trying to restore what had been there in the past. These ecosystems evolved over millions of years, and essentially in a very recent time frame, humans have come and in a lot of instances, unfortunately, degraded habitats and resulted in species level decline. So we're trying to restore what used to be out there. We're not moving mussels or snails or fish somewhere where they didn't used to be. And actually one of the benefits of having such a great natural history collection is that we can see where these organisms used to be. And so you might be asking, well, how do you know they used to be there? Well, we look at those specimens that were collected in the 1800s and in the 20th century, and then we can see, okay, this species used to be there. We've been out to those sites recently. It's not there anymore. We think the habitat maybe is restored. Maybe there's been some physical restoration. Maybe the water quality is improved with land use changes. And we're going to maybe target that system as a place to put a species that is still around in other places back in the area where it used to be. And by doing that, we can hopefully make the ecosystem look like it did before humans caused habitat degradation.
Is your work in this area mostly in Ohio, or is it happening elsewhere?
Actually, a lot of my work has been in the southeastern United States. So I actually started here at Ohio State in August, and before that I was working in the southeast with U.S. Fish and Wildlife Service and Auburn University. So a lot of my research did happen in the southeastern United States, and that's where a lot of it will likely continue. But one of the things that's really exciting about moving to a new place, for me, like Ohio, is being able to expand my research and focus on some really pertinent questions in Ohio. And I think there's some really exciting work that needs to be done on freshwater mollusks diversity here in Ohio.
You'd said before we started recording that a common theme, at least in work that you've done in the past, is that most common knowledge about freshwater snails is wrong.
Yes.
What do you mean by that?
Yeah, so, this is the case for a lot of understudied organisms. Since they're understudied, maybe one group gets a really detailed study. Maybe somebody's very interested in, we'll use apple snails, for example. And what happens sometimes is if somebody finds, something about the biology of apple snails, since a bunch of other snails haven't been studied, sometimes they'll get extrapolated and, repeated as like, oh, well, this happens in all freshwater snails. But freshwater snails are a really diverse group. They've been on this planet for hundreds of millions of years. And so, what happens in the biology of one group of snails might be very different than the biology of another group of snails. So one of the things that I realized very early on is some of the common knowledge, some of the things that have been said and repeated both anecdotally and in the peer-reviewed literature about the snails that I really study. It's a family called Pleuriseridae. They're here in Ohio, very diverse in the eastern United States. A lot of things people have been saying was wrong. For example, it had been said in the literature a few times that these individuals, the females would lay eggs once and then they would die. It's a common life history strategy out of nature. For example, we know octopuses do that. But we were pretty sure this was wrong. In fact, we knew it was wrong because I was working with the Alabama Department of Conservation and Natural Resources looking at captive propagation, and they had females that they knew had been laying eggs multiple years because they would keep the age cohort separate. And every year, for about 6 years, this species, these females would lay eggs. So we knew that was wrong. And so we published on that. We also were doing an anatomical study where we were looking at reproductive anatomy. And we started looking at snails that we had collected in August, and we were pretty confident they were females, but we couldn't find any evidence of sperm storage, even though all of the literature and anecdotes had suggested that these snails store sperm for a long time. Well, we did histology, and we realized that was simply wrong. We think now, based on what we've seen in captivity, although watching actual mating is a little difficult in these small snails, but we think they actually mate, and then they lay eggs about 2 weeks later, and then the eggs would hatch about 2 weeks after that. And they don't overwinter sperm. They're incapable of doing it. And so things like that are some of the common knowledge that we've kind of really tried to examine and assess.
I'm sitting here trying to understand what explains this misunderstanding about this. It sounds like it's been persisting for a long time. What explains that?
I mean, I really think it's just how few people are studying it, right? So, if we think of, I'll use birds and reptiles as an example. There's a lot of research, and that's not to say there isn't more research needed on those organisms, but there's simply more people studying birds than there are freshwater snails. And so, if somebody, says something about a bird species, because there's so many people studying it, it's probably going to find out really quickly whether, that's right, or that also happens in this other organism, or it doesn't happen in this other species. And that's just not the case for freshwater snails. Somebody may say something about a species, and they might think it's right, and then somebody kind of just, repeats it for this other species, or simply because there's no data, it's the best we have to work with. So we say, okay, well, this species is doing one thing, this closely related species must be doing it as well. And since there's no one really studying those organisms, maybe years or decades before somebody actually assesses that and says, okay, well, that is true. This species is doing that. Or actually, that's not the case. These species aren't, for example, overwintering sperm or mating in the fall and then laying eggs in the spring. So I think it really just comes down to how few people are studying these organisms. And, that's one of the things that really fascinates me. That's one of the things that I really like training students in, because, it's a group that needs more researchers and needs more advocates, and that's one of the reasons I'm really passionate about them.
You just drew attention to the fact that you came to Ohio State just a few months ago from Alabama. What drew you to Ohio State? What drew you to the Museum of Biological Diversity?
I mean, really the opportunity to lead a world-class natural history collection was, something I just couldn't pass up. I really am passionate about organismal biology, and research collections are really important. They, as I mentioned, they document not only our shared natural history, what used to be here, but they also serve as a resource for documenting current biodiversity and being able to deposit our research vouchers in these collections. So, being able to be the director and, think about some strategic leadership opportunities at such a world-class institution was really something that compelled me to want to come to Ohio State and be the director of the Museum of Biological Diversity.
A moment or two ago, you said that the museum doesn't do public exhibitions, for instance, but that you're starting to do more work in the community. Like what?
Yeah, so, we actually have an open house planned on March 28th. We're really excited about that.
March 28th, 2026.
Yes, March 28th to 2026. And this was something that had been happening for many years, and it stopped in 2020 with COVID, and then it never really started back up. And I think, it used to be a very large and one of the premier outreach events in the College of Arts and Sciences, and we're really looking forward to restarting that and again becoming a premier outreach and public event for the College of Arts and Sciences and specifically the museum. We're also working and trying to get some of our stuff back into the community, and we're kind of thinking about how we can engage with the community more often. I think when people hear about a natural history collection or a natural history museum, they might think of the Smithsonian, if you've ever been to see and seen that. We don't have an exhibit like that, but we have the research specimens that the Smithsonian has. Maybe not quite as big in some areas, but that's part of our thing. And so we're thinking about how we can use our collections to design exhibits that can, small exhibits that we can get in the community, participate in other outreach events, and really just excite people about what we have here at Ohio State and also in Ohio.
Where's the museum located?
Yeah, it's on Kinnear Road, so it's in West Campus, so off the main campus, kind of in a nondescript building. And that kind of comes with the territory of not having a public exhibit, but it should be pretty decked out for the open house in March.
So we were talking about genomic data and computational methods, computational biology. What are some of the new technologies that you're bringing to research and interpretation of these collections?
I mean, I think one of the things that really excites me with some of the genomic tools is being able to use specimens that were collected, 25, 50, 100 years ago to do, actually whole genome sequencing on species that might be extinct. So that's something that we're really excited about pursuing. We can make small drills into a shell material, for example, to get genetic material, and it's very degraded DNA. It's fragmented, but with new genomic technologies, we can potentially sequence the DNA of extirpated and extinct populations, and also get a better sense potentially of genetic diversity through time. And we can do that with mollusks, but we can also do that with our other collections in the museum. So that's definitely one technology that we're excited about. With genomic data, we can also better assess genetic diversity and kind of, like as I mentioned earlier, demographic history. So getting a more fine scale sense of what's really going on in the populations that are still out there, and maybe what happened in the past, is something that we're really excited about. And we try to stay on top of the latest genomic sequencing technologies for a very long time. Well, the technologies haven't been around for that long, but for as long as they've been around, a lot of the sequencing is very short reads. So you might sequence 100 base pairs, so an A, C, G, or T. 100 base pairs.
DNA.
Yeah, the DNA. So you're sequencing short fragments. And then it can be difficult, going back to the computational biology, to kind of assemble that into meaningful stretches of what the genome actually looks like. And so there have been recent advances in what's called long read technology. So sequencing 500 plus 10,000 base pair sequences of DNA at once, and that can help us better assemble the genome of these organisms, and we can then look at things like structural variants in the genome to get a better sense of kind of what's going on and how these organisms have evolved, but also how changes in habitat and changes in conservation status have affected their genomic compositions.
I pay a little bit of attention to CRISPR technology, gene editing technology. Has that technology worked its way into organismal biology?
I mean, it certainly has in some fields. It's not a technology that we've used in my research, but you could certainly imagine potentially down the road it being used for conservation, especially if there's a trait that we know, a genetic trait, a gene that's very beneficial to organisms when we think about captive propagation. This is very hypothetical, but you could certainly think up ways that CRISPR and gene editing technologies could improve conservation outcomes. And then, of course, right now, and this is not something my lab does, but there's some movements to do de-extinction of different organisms, and that's not something my lab is involved with. And I think
Jurassic Park. Right?
Yeah, and it's kind of difficult when you're whether that's really the best focus of our resources. Because there's a dichotomy, right? We could focus on conserving what we still have out there, which I would argue is actually a little easier than trying to make something come back to life. Or we could focus on bringing something that kind of looks like we're never going to de-extinct something completely, but maybe it looks like, for example, the dire wolf hit some headlines recently, and so that is certainly a technology that who knows where things go. With genomic stuff, things move very, very quickly. So that is an emerging technology. I'm not aware of anyone using that at the museum or even at Ohio State, but you never know where things might go. And unfortunately, we're in a biodiversity crisis, and so we are getting to a point where I don't think any idea can be shot down just out of hand when it comes to trying to prevent extinctions.
I know that part of the mission of the museum is to train the next generation of biodiversity scientists. Yeah. How are students engaging with the collections? What opportunities do you see for future workforce development?
I mean, advising, educating students is one of the most enjoyable parts of my job. So each of the research labs there have graduate students working in their labs. So we have a lot of graduate students working in our collections, whether that's depositing specimens that maybe they collect for their own research, or using the historical documentation of organismal diversity as part of their research. There's a big part of that. We also have undergraduate researchers working at the museum. We'll often also have times we'll have classes come by the museum to kind of educate them about a specific organismal group. Maybe that's plants, and give them a really specific tour of the herbarium, or just kind of a broader scale tour looking at kind of the whole museum, but also how museums work and how a research collection might differ from a very public-facing museum. And then we have we'll have student groups come through, like the zoo. I gave a tour with a colleague just the other day for the zoology club, and just kind of showed them our specimens. And it's always really awesome to see students and young people get excited about the natural world. And I think my experience has been that people are really interested in this sort of thing. They're really interested in what's here in Ohio, and people generally like protecting what they have and what is unique to a place like Ohio. So we like emphasizing, at least I like emphasizing when I give tours, the things that are unique about Ohio, because a lot of the people who come through the museum are from Ohio.
Your site emphasizes creating an environment where all the lab members feel empowered.
Mm-hmm.
How do you cultivate that culture? And why do you think it's important to create such an environment like that?
Well, I think it's really important that students have some ownership in their learning, generally because I think that kind of allows them to learn more. It excites them a little bit more when they have some kind of shared ownership and they get to choose what they want to do. I mean, in terms of creating an environment where everybody contributes, I mean, I think a big part of that is making sure everyone has a voice when we have meetings, when we do research. I really encourage my students to come up with research ideas on their own, with feedback from me and others and their collaborators. But I really like students seeing how they can develop their research skills, and a lot of that is coming up with new research ideas. That's a huge part of research. And so, one of the things I try to tell my students is like, hey, did you want me to do this analysis of this? And I usually like, well, it's not so much what I want you to do is like, does it make sense? I mean, I might say something boneheaded one day, and like, it's good for my students to be like, hey, Nathan, that doesn't make any sense. And I encourage that. I want to make sure that my students are thinking through the method and not just doing it because, that was in some other publication or I had mentioned it, in a lab meeting, for example. So really encouraging my students to do things because it interests them, because it makes sense to them, it's scientifically valid, not just because, that's what this paper did, or that's what Nathan mentioned I might want to do, because, again, that might be how we start repeating some common knowledge that's not true. And certainly, I think of myself as an expert, but I certainly don't know everything, and, I like learning from my students too. And so that's why I really encourage them to develop their own voice as a scientist, their own ideas as a scientist. I think that's really important, especially for graduate students who are, learning how to do research to go out in the field and make an impact.
Where did your interest in evolutionary biology come from? Did you know when you were 4 years old you were going to be a biologist?
I mean, not really. I thought for a long time that I was going to be a high school teacher. When I went to college,
Science teacher?
Yeah, science teacher, biology teacher potentially. I mean, I was interested in biology. I thought conservation was really important. I don't know exactly when I developed that, but you spend a lot of time outdoors as a kid, so it probably just kind of came from that. And I decided when I got to college, I went to school at a small liberal arts college in Northeast Missouri called Truman State University, and I did some undergraduate research, and I got really interested in organismal and evolutionary biology. Really, I'm not sure I really knew that you could do research as a career. I mean, I knew there were professors, and I knew that they taught classes, but I don't think I had a sense of the kind of research that's going on at a lot of universities. And so that's when I got excited about it. I went off to grad school. I decided I wanted to pursue a PhD. I did that at the University of Alabama. And that's where I really got interested in freshwater snails. I wasn't so set on, when I got to grad school, an organismal type. I mean, I actually joined a fish lab for grad school. But I shortly after joining the lab and getting to Alabama, met some people with the Alabama Department of Conservation as a resource. In fact, one of them I still work with, Paul Johnson, and he's an expert. He knows much more than me on these freshwater mollusks and snails. And I met with him, and it was very clear that there was a need to do good genetics research, good evolutionary biology research, and on these organisms. And his enthusiasm was really infectious, and that got me involved. And that that actually speaks to how important good mentors are in this field. And I've been very fortunate to have good mentors throughout my career.
What would you say has been the most exciting research find of your career thus far?
Well, one of the things that got me super excited and really convinced me that this was work worth doing is when we found species in the wild that people thought were extinct. So, and this kind of goes back to that common knowledge issue with these snails. They're, again, very understudied, not many people looking for them. But there were a handful of species that had been declared extinct, and some of them probably are. But a few of them we found out weren't. So, for example, in 2011, when I was still a grad student, this really invigorated me as a grad student. There was a species, the oblong rock snail, that is in the Cobb River in Alabama. It was the only snail species that had gone extinct in the Cobb River. This is one of the most unmodified river systems in the southeast. I asked colleagues who had been doing this much longer than I had, why did this species go extinct? What about it, caused it to go extinct? And I never really got a satisfying answer. And so after doing some work actually at a natural history collection, but down at the University of Florida, I kind of had a sense of like, okay, this is where the species was most abundant or most commonly found historically. We should go look for it. Like, when was the last time? And so I gathered up some friends, and we were like, okay, let's do a canoe trip down the Cahaba, and we'll enjoy our Sunday. We'll also look for the snail. We put our canoes and kayaks in, and I would say about 5 minutes into the trip, we find the snail. And so it was great. I was like, I don't think my friends believed me at first. I was like, guys, we found it. I think they're like, okay, we just started. You didn't just find it. And sure enough, it was right there. It'd been overlooked. Not the easiest place to access. It was kind of about as far from Birmingham as you can get downstream, but still kind of, be above another kind of moderately sized city, Centerville. And so difficult to sample area, but I just don't think people had looked for it. And so ever since doing that, it's kind of like, okay, we might not know where these species are as well as we think we do. And so then back in 2024, I think it was, 2023, 2024, we actually found another species that people thought had been extinct in Mississippi. And that was an example of some colleagues were doing mussel research. So, mussels I think are understudied, but compared to snails, they're much better studied. And I told him, I was like, hey, I know you're searching for mussels, but keep an eye out for some snails. There's a species that is extinct in the Big Black River, but I think it might still be out there. I don't think we've looked hard enough. And about 6 months after they did the survey, I got an email from one of my colleagues. I was like, oh, hey, I forgot to mention we did find the shell. It was a dead shell, but and I was like, well, that's it. It's got to still be out there. And he was kind of like, well, this river, the shell's kind of preserved in the silt. I was like, I've seen a lot of these shells. That's not a 60-year-old shell. We got to go look for it. So we look at the map, and we're like, well, geez, that's a 2-hour jet boat ride from the nearest boat launch. I mean, this is a remote part of Mississippi. And, how are we going to get there when the water's low, which is the best time to sample these things? And saw on the map that there was a wedding venue or an event venue nearby. And we're like, well, let's call up the event venue and see if they'll just access our land. And sometimes you hear stories about people not wanting like biologists tramping on their land. And so we asked a colleague who does with Fish and Wildlife Service, does a lot of work with landowners, if they could call the person and kind of put on the sales pitch. And they were like, oh yeah, come on by, come on through, we'll let you access their land. And so we went to that spot. My colleagues got in the water, and I mean, literally millions of them at that site. Very easy to sample. We did some very quick estimates to understand how many there were. And we think there were probably close to 1 million of the individuals. They're very locally abundant, even when they're very rare. And so that's really exciting. And what's the most exciting part about finding a species that we thought was extinct is that then we can actually try to conserve it. We can't conserve something generally that's already gone extinct. But once we find it, then we actually have an ability to protect what's still there and potentially do captive propagation or reintroduction to put it in places where it used to be but aren't anymore.
So as director, what are your aspirations for the Museum of Biological Diversity over, say, the next decade?
Well, I think it's two things. On the research side, I want to see our collections build. I want to see, I want this collection to be, the museum to be somewhere where people deposit their vouchers. And certainly we have researchers now that deposit their stuff. But, what I'd like to see is, if you're doing research here at The Ohio State University and you have organismal voucher specimens, we want to voucher them at the museum. We want this to be a repository and a resource for the researchers here at Ohio State. So that's on the research side, really building our collections even more. On the public engagement side, I really want to see us do more with that. I think restarting the open house is a big part of that, and I expect it to be a really great annual event. But I really want to think, down the road strategically about how we can get our science, get our collections, in front of the community. People in the Ohio State community, but also in Columbus and Ohio and the Midwest more broadly. And so, one of the things that I hear in the community when I've talked to people, met people since I've moved here is, I'll mention the museum, like, oh, that open house, are you guys going to be doing that? That was great. I was like, yes, now we actually are. And so, people who've experienced the museum have been engaged and have been excited about it. And so what I want to see is that excitement build, and I want people to not talk about something that happened 6 years ago. I want to hear people talk about, oh, that open house last year, that thing y'all did, earlier this year was exceptional. And so I think building the research collection and building the public engagement, when I retire, if we see that having been built, we have more specimens, maybe build our endowments as well, that'd be great. And also, having made an impact in the community, I'll be very happy.
Nathan Whelan, thank you.
Thank you for having me.
Voices of Excellence is produced and recorded at the Ohio State University College of Arts and Sciences Marketing and Communications Studio. More information about the podcast and our guests can be found at go.osu.edu/voices. Voices of Excellence is produced by Doug Dangler. I'm Jen Farmer.